Hello, today's topic of discussion is SDS page. If you are new to this channel, consider subscribing and click on the bell icon to get notified. So SDS page is the sodium Dodecyl sulfate polyacrylamide gel electrophoresis. It is an electrophoretic technique used for the separation of proteins. The gel matrix used for separation is polyacrylamide gel, and the gel is prepared by cross-linking acrylamide and bisacrylamide by free radical catalysis. So APS and TEMED is used for the polymerization of acrylamide. The pore size of the polyacrylamide gel can be controlled by adjusting the concentration of acrylamide concentration. So based on the concentration used, the pore size can be varied. The polyacrylamide gel electrophoresis system consists of two types of gel. One is the stacking gel and the other is the separating gel or the resolving gel. Stacking gel is usually of 1 cm long, 1 to 2 cm long, whereas the separating gel usually is 10 to 12 cm long. The protein can be loaded onto the stacking gel, the wells in the stacking gel, and electric field is applied. The protein will move through the stacking gel and enters the separating gel, and in the separating gel the proteins will get separated based on the size of the protein. So in SDS page, the SDS imparts a negative charge onto the protein. So the proteins will be uniformly charged, negatively charged, and under the influence of electric field they separate only based on the molecular weight. The sample buffer or the denaturation buffer used for preparing the samples includes TETRamethylethylenediamine, beta-mercaptoethanol, SDS, glycerol and bromophenol blue. So these are the components in the sample buffer. So these TETRamethylethylenediamine provide chloride ions, and the beta-mercaptoethanol is used for as a reducing agent which denatures the disulfide bonds of the protein. SDS also denatures protein, and also it gives the protein a net negative charge. SDS binds to the amino acid side chains and gives the negative charge. So SDS is a detergent, anionic detergent, and upon binding to the protein gives a net negative charge to the protein. The glycerol used in the buffer provide density or the viscosity to keep the sample in well. And the bromophenol blue is used as a tracking dye for tracking the migration of protein on the gel. So here you can see the protein has different bonds. It has hydrogen bond, ionic bond and disulfide bridges. So these need to be disrupted to run the protein uniformly on the gel. For that, SDS is used, as well as the disulfide bond breaking reducing agent beta-mercaptoethanol is also used. SDS binds approximately 1.4 gram per gram of protein. That's why it gives the protein a negative charge, net negative charge. And the reducing agent beta-mercaptoethanol, they disrupt the disulfide bonds of the protein. So the SDS page runs as a discontinuous buffer system. It has three major charged species. They are chloride ions, the SDS protein complex, and the glycinate ions. The chloride ion, which has the highest mobility, and the SDS protein complex moves, and finally the glycinate, which at 6.8 pH runs as the slowest. So in the stacking gel, the protein gets sandwiched between the chloride ions and the glycinate ions, and they stack together uniformly. So once the electric power is applied, the chloride ion leads the way, followed by the protein, and glycinate essentially wedges the protein between itself and the chloride ion. So this makes a tight stack of the protein into tight thin starting zone, and that's why the pH 6.8 gel is called as the stacking gel. It helps stacking the protein together. Once the complex hit the higher pH running gel, that is the pH 8.8 or the separating gel, the glycinate ion becomes more fully ionized and its mobility increases, and it overtakes the protein SDS complex and follows the chloride ions, and these two species leave the proteins behind. and the proteins separate based on the molecular weight. The protein keep moving towards the anode, all with the same electrical mobility. Since the presence of SDS, it has identical charge per unit mass. However, without following charge species to keep them stacked together, they are now free to start separating based on their relative sizes. So the important thing to consider here is that the presence of two different buffer system and the presence of trisglycine buffer as a running buffer. The glycine, the amino acid is a citraionic form in its isoelectric pH, and at lower pH it becomes positively charged, and at higher pH it becomes negatively charged. So that's the reason when it enters to 8.8, that becomes fully ionized and moves faster, leaving the protein behind. And the protein will get separated truly based on the molecular weight due to the sieving nature of acrylamide. Generally, the stacking gel is of high porosity. That means generally it is prepared using 5% acrylamide gel, and it is more porous, so there is no sieving effect in the stacking gel. Whereas separating gel is prepared based on the molecular weight you wanted to separate, and generally it is 10 to 15%. So there the protein separation or the sieving effect occurs. So the protein will get separated based on the molecular weight. So here is the protein gel after staining. So here you can see there are different bands of protein, and all of them separated based on the molecular weight. Hope you are clear with this topic. If you like the video, please click the like button and share it with your friends. Thank you.